This is the Week 2 overview, upstream and downstream processing. The objective of biomanufacturing is to maximize the quantity and quality of the products that are produced. The bioprocessing workflow is divided into the upstream process and the downstream process. The upstream process focuses on ensuring quantity and the supply of all of the components that go into the bioreactor and the biologic production. The downstream process focuses on optimizing quantity and quality of the desired product from the fermentation or cell culture. What is a bioprocess? This is the application of natural or genetically manipulated whole cells, tissues, organs for the production of an industrial or medically significant product. Some examples of products of a bioprocess are organic acids or alcohols, flavors and fragrances, DNA for gene therapy, antibiotics, proteins such as monoclonal antibodies, interleukins, interferons, various enzymes, hormones such as insulin or human growth factor. The objectives of bioprocessing are to apply and optimize natural or artificial biologic systems by manipulation of cells and/or their environment to produce the desired product at the required quality. Genetic engineering is a critical pathway to achieve this objective. The model systems that are currently used are viruses, prokaryotic cells such as bacteria, or eukaryotes such as animal cells, plant cells, whole plants, whole animals, or transgenic animals. What are the steps in the bioprocess design? First is the product definition. This is defining the product specifications and defining the analytic needs. Second is selection of an optimal synthetic technology, including determining what is the optimal model system. Third, we create the process flow diagram. Fourth, we identify the material and energy balances to calculate the cost. That includes the materials, the reagents, and consumables, as well as equipment. The fifth, we identify the economic and quantity hotspots. What is the application and what would be the ultimate goal of our product? Sixth, assessing the profitability and risk of the process. And seventh is to create an R&D agenda for how to optimize the product quality and quantity. What are the design criteria? These things can include the concentration that is produced by your model system, the productivity, that includes the volumetric as well as specificity of product production, the yield and conversion to your product, and perhaps most important is quality, and that means the purity, the sequence. Is the sequence accurate? Is it glycosylated? Does it have post-translational modifications? Are there foldings of these proteins? What is the biologic activity measured both in vitro as well as in vivo? This diagram shows the upstream and downstream process and how they're interrelated. The upstream process includes the selection of our product, the selection of the producer organism, the optimization of the production or the media allowing the production of our product, the initial small-scale setup of our bioreactor, and the components that lead to how to scale up the production of our product for optimal production. And at this bottom of the screen, we then show that this last step then leads to the downstream process. Once we have the product in hand, how do we purify it and finish it to a point where it can then be utilized either industrially or clinically, either in a clinical trial or for industrial applications, to a point where the purity is adequate for the utilization for these processes. The upstream process. The upstream process is defined as the entire process from early cell isolation and cultivation to cell banking and culture expansion until the termination of the culture and collection of the live cell batch. Upstream processing refers to the initial step in which the cells or microbes are grown in bioreactors and involves the steps related to the inoculum development, media development, improvement of the inoculum by genetic engineering, optimization of the growth kinetics, such that the product development is optimized. Upstream processing can be divided into three primary areas. First, the producer organism, that is obtaining a suitable microorganism or cell type, strain improvement to increase the productivity and yield, maintenance of strain purity, and preparation of a suitable inoculum. The next is the fermentation media or culture media, as well as the fermentation process, i.e. those steps involved in the actual fermentation process. The most important step in bioprocessing is the choice of the production cell line. The choice of the cell line or microorganism used profoundly affects selection of the bioreactor, the downstream processing, feeding regimen, as well as the scale of production. Here we show three different organisms or cell lines that one may choose from. For example, bacterial cells. It has the advantage of genetic ease. It has single molecule DNA that's been highly sequenced. It has high productivity. It's resistant to the shear force and osmotic pressure that's associated with the bioreactor. These are immortal. However, they have certain negatives. They are poor secretors. They have little or no glycosylation or post-translational modifications. One can use yeast. They have the advantage of they can grow at high cell concentrations. They're high productivity. They're good secretors. They exhibit some post-translational modifications, and glyco-engineered strains are available. The disadvantage is that the glycosylation is exhibited as a non-mammalian glycosylation and non-mammalian post-translational modifications. And there's also an added complexity of genetic manipulation. One can use mammalian cells. These have the advantage of producing human-like proteins. They can be engineered to be high secretors, and they can correctly construct and are biologically active. Their disadvantage is they generally have slow growth rates. They only grow to low cell densities. As a result, they have low productivity, and they're extremely sensitive to the shear force of the bioreactor and the osmotic pressures. They are substrate and product sensitive. They also can be affected by apoptosis, and their cells age. A significant issue of having proteins as the product of the bioprocess is that proteins and their activity are highly sensitive to the protein structure. Proteins, we can generally think of them as a sequence of amino acids. This is their primary structure. Equally important is their secondary structure. What kinds of shapes? Are they folded? Are they twisted? The most common shapes are alpha helix and beta sheets. An additional importance is their tertiary structure or three-dimensional structure. How they're formed with their secondary structures are cross-linked. And then there's their quaternary structures, or their unique globular three-dimensional complexes made of multiple polypeptides. All of these together can determine the biologic activity of the protein that's produced. The second critical step in the upstream process is the selection and the type of bioreactor. And this depends on a number of factors. First, are the cells anchorage dependent or suspension adapted? There's mixing considerations in terms of homogeneous conditions, the absence of nutrients, and temperature gradients. There's the mass transfer, cell density. How dense can some of these cells go? Shear resistance. As we showed, bacteria are extremely resistant to shear force of the bioreactor, whereas mammalian cells are very sensitive. Their validation issues. Can these be used and reproduced in various conditions and then scaled? When brought up to scale, do they maintain these functions? The downstream process. The downstream process involves the recovery and purification of biosynthetic products from natural sources such as animal or plant tissue or fermentation broth. This is an essential step for the manufacture of pharmaceuticals, antibodies, vaccines, enzymes for diagnostics and industrial applications, and natural fragrance and flavor compounds. Downstream processing refers to the separation and/or purification of biological products on a large scale. Downstream processing leads to the purified product suitable for a specific use. This figure shows the need for downstream processing as well as the challenge. Here we show the bioreactor. The input for the bioreactor are cells, cell culture media, and media supplements. In the effluent, in addition to your specific product, we have cells. That includes the intact cells, cell debris, host cell proteins, potentially viruses, DNA, lipids, and other cell contaminants, potentially endotoxins. You have the media supplements, particularly from those derived from the bovine serum proteins. You have contaminants from the cell culture media. These are amino acids, inorganic salts, vitamins, glucose, and other organics. Downstream processing is divided into 4 steps. First, removal of insolubles is the first step and involves the capture of the product of a solute in a particle-free liquid, such as separation of cells, cell debris, and other particulate matter is achieved. There's product isolation. This is the removal of those components whose properties vary markedly from that of the desired product. In most cases, water is the primary impurity, and the isolation steps are designed to remove it. Third is product purification. This is performed to separate contaminants that resemble the product very closely in terms of physical and chemical properties. This stage contributes to a significant fraction of the entire downstream processing cost. And last is product polishing. This describes the final processing steps which the end product is packaged in the form that is stable and easily transportable and convenient. Purification techniques. These can include filtration, which is a separation based on molecular size, precipitation, liquid-liquid two-phase separation, chromatography. This is the most commonly utilized technique, and this can include size exclusion, gel filtration, ion exchange, hydrophobic interactions, reverse phase chromatography, hydroxyapatite, and a variety of affinity-based chromatography techniques. In summary, bioprocessing is an integrated process designed to take all parts into account to provide the quantity and quality of product required in the most efficient and cost-effective manner. Upstream process design accounts for optimization of the product production, while the downstream process is critical for isolation and preparation of the product for